Hello everyone, welcome to my YouTube channel. So here today we are going to be looking at a full paper for biology, which is 2025. So this paper is very good for your revision and for your knowledge of how you can apply certain concepts. If you are doing any NS program, this paper is for you. So we have the first question. It says that you have an individual who is totally heterozygous for two genes that are not linked, that is, not on the same chromosome. One gene is for ESIs, HH, and so these are the letters. Then we are told also that the other gene is for baggy eyes, which is Q like that, for baggy eyes, and whereas two small letters we have the normal eyes. If you test cross this individual, what will be the resulting phenotype? So to find out the resulting phenotype, what we are going to get is we are going to cross them. Now the key term for this question is you have to be very careful. They have said if you test cross this individual. A test cross simply means to say we are going to fuse this individual with someone who is totally homozygous recessive, meaning to say they have recessive genes. Now, just some background information. Where is the test cross used? So the test cross is used to mostly identify the genotype of the parents. For example, if a parent, let's say if a parent is tall, it is possible that the parent has both genes of tallness like that, or they have both of these are tall. According to genetics, both of these are tall. So therefore, test cross can be used to fuse these two parents with someone who has two genes which are totally recessive, meaning to say they have small letters like that. Now you will find that as you cross, you will find that this T and this T will end up crossing each other. You will find something like this. So there would be an outcome which is short. Now for this other one, there would be an outcome that is short. Now if the parent has got both letters that are big letters, there is no outcome that can be able to more like yield. So here we could have T, even here we could have still T. We are always having a big letter. So if the outcome is always showing you, if there is any outcome that is having two small letters, then that means to say the phenotype of the parent is homozygous dominant, meaning to say we have the tau trait, which is that one. Now in real life, it's not that they show the exact traits for tallness. No. For example, if you want to determine the genotype of a dog, you get a smaller dog. Then with that smaller dog, you are going to fuse it with a small and a big dog. So a big dog you don't know, then you have the small dog, which you know to say for this to be small, it has to mean that it has got two small letters. But for this one, you don't know. So as they continue to more like mate, they will produce offsprings that might be sometimes small. Okay, sometimes let's say big. If in all the outcomes, all the offsprings are big, that means to say this parent has got big letters. But if there is any dog that comes out to be small, it means to say this dog is big, but it has to have this type of a genotype. So how do we do dihybrid crosses for this? Now remember, these are not linked. It simply means to say someone can be, let's say for example, traits that are not linked. Someone can be tall at the same time that person can have, let's say, blue eyes, or someone can be short and have blue eyes. They are not linked. Someone can be tall but have the black eyes. So that means to say these are not linked. The possibility of having one trait is totally independent with the probability of having another. if this is what we have, we are going to say you have an individual who is totally heterozygous, so this is a test cross. Now, the last part was saying that if you test cross this individual, what will be the resulting phenotype? So a test cross already requires what we are going to have is what do we have? We have these letters. So if this individual is heterozygous, it means to say they have the trait for, let's say, which characteristic is this. So we have for ESIs, they have to have the genotype. So we are getting the genotype of this person. Since it's heterozygous, it means they have to have a big letter H and a small letter H, like that. Then we have for eyes, we are going to have the genotype for eyes. For eyes genotype, we are going to have the Q, like that, and the small q, because this individual is heterozygous, meaning there is a small letter and a big letter, like that. Then if we have that type of a situation, what are we going to have? So what we are going to have is that, let's come up with a Punnett square where we are going to put these traits. So this H and the small h there. Then for this other part, we are going to have something like this. We have our Q and our little q, like that. Then we have our H, our Q, our H, our Q. Then we have our H and our Q, like that. Let's begin with our H. H, big Q. So we are just combining. We have our H, small q, like that. So this is the other outcome, like that. So now when you draw your phenotype, the genotype outcome is that we have our HQ, like that. We have our H, we are here, we write our HQ. So we have our H big Q, we have our H small q, like that. So this same thing, we are going to now look at the test cross individual. So this test cross individual, we are going to have, this person should have both traits, they should be recessive. That means to say for ESIs, this person should have two small letters for H. Then for I's, we should have, this individual should have two small letters. So the test cross always has small letters. So we are going to have we are going to fuse them like that. We have our H there, we have our H there. Then on this other side, we have our Q and our Q, like that. So we are going to get something like this. So our HQ, so we have our HQ, we have our HQ, we have our HQ, we have our HQ, like that. So the outcome here is our HQ. So this is the genotype. So this is the genotype. So the genotype is equal to, we have our HQ, we have our HQ, we have our HQ, we have our HQ. From there, come up with a 16 Punnett square. So when we get our 16 Punnett square, we are going to put this there. So we have our HQ, like that. So we are going to get our HQ, put it here, HQ, H small q, small h, big Q, small h, small q. We have obtained that. Then this side we are going to put this. So we have our HQ, we have our HQ, we have our HQ, HQ, like that. We are going to combine, so our H and H, so we have H small h, Q small q. Same here, H small h, big Q, little q. H small h, big Q, little q. H small h, big Q, little q. We have our H small h, small q, small q. Our H, small h, little q, little q. We have our H, small h, little q, little q. Our H, small h, small h. Then we have our little q, small h, big Q, little q. So we are just combining them. Then we have our H like that, our H, then we have our big Q, little q. We have our H, big Q, little q, H, big Q, little q. So we are just combining H, Q like that. Then here we have our small letters H, small q, like that. Then here we also have our H, as you can see, H, small q, H, small q, our H, it's just our H, small q, like that. So this is what we have for that problem. Now after we are done with this, we are done with our 16 Punnett square, then we are just going to identify. So how you identify? You are going to say, number one, what type of ears are you given? You can say big ears, according to the question, big ears and baggy eyes. Number one. Number two, you have your big ears. So these are the possible outcome. Big ears and normal eyes. Then if these are the possible outcomes, then you are going to switch to small ears and baggy eyes. Last but not the least, we have small ears and small eyes. Sorry, not small eyes, but they were using the word normal eyes like that. Then what we are going to do is we are going to count. So from genetics, we know how we count. So counting, we are going to say for big ears and baggy eyes, we are looking for anywhere where there is a big H, at least one, and at least one big Q. So if we have, let's say, this and this, so this is number one, number two, number three, number four, as long as we have at least one big letter Q and one big letter Q and one big letter H. That means to say we can pick that as one. Now here we have one big letter Q, we have one big letter H, but we do not have any other big letter Q. As you can see, even here we need at least one H big letter and at least one big letter Q for us to have to conclude to say big ears and baggy eyes. So here we are going to have 4 like that. Then when we move to big ears and normal eyes, so big ears, we are looking at big letter H and at least 2 small letter Qs, because normal eyes, according to this, is recessive. It will not show if there is a big letter. So just as you have seen, the question says for baggy eyes, it's big letter Q, 2 big letter Qs, or Q little q. So that means to say for normal eyes, you have to have 2 small letters of Q. So this is going to give us, we have number 1, number 2. This is where you have at least 1 big letter H but 2 letters of Q. So we have this. So we are going to say this is a 4 there. Then here we are going to have our small ears. That means to say we expect 2 letters of H, baggy eyes, at least 1 Q. So where we have that, it's number 1, 2 small letters but at least 1 Q, 1 big letter Q. So we have 4 there, so we can count as 4 of them. So we have 4 like that. Then here we have small ears, meaning 2 small letters of H, and normal eyes means 2 small letters of Q according to this. So here we are just remaining with number 1, number 2, number 3, and number 4 like that. So we can have something like this. We have 4 like that. So the ratio is a 1 to 1 to 1 to 1, which is the same as a 4 to 4, 4 to 4 to 4 to 4, which when reduced, if you divide 4 throughout, you're going to have a 1 to 1 to 1. Let's move on to the next question. We are told that tomato plants are produced by the action of a dominant allele D and a dwarf plant by its recessive D. Hairy stems are produced by the dominant gene B and the hairless by the recessive gene B, small letter b. If one heterozygous for both traits, tomato is test crossed. So test cross means you use small letters. Therefore, states the resultant phenotypic ratio. So phenotypic ratio, we are going to do the same thing. So when we do the same thing, we are going to get something like this. So this tomato, what have we been told? So if one heterozygous tomato, meaning to say it has got number 1 for the tall tomato, it is represented for the tall plant, it is represented by big letter D and the small letter d, since it's heterozygous, meaning to say both are present. Then when we look at the stems, we have hairy stems is represented by big B, small b like that. We are going to do our 4 Punnett square first to fuse them. What are we going to get? So we have our D, small letter d, we have our B, small letter b like that, small letter b like that. Then when we do our cross, we are going to have D, small b, we have our D, small b, we are going to have our D, big B, then we have our D, small b like that. So this is what we have for that one. So this is what we have for that one. Now when we have done that, so the outcomes are our D, small letter b, we have our D, small letter b, our D, big letter B, our B, our D, small letter like that. So what we are going to have is, after we do this, we are going to use now the test cross. The test cross plant has to have small letters of D and small letters of B like that. So we are going to do the Punnett square like that. When we do our Punnett square, we have our D like that. Then here we have our D, small letter d, we have our B this side. So we can fuse them by saying this is D, B, D, B. We are just combining them. We are here, D, B. We have our D, B like that. Therefore, when we do each and everything that we need to do there, what are we going to have? What are we going to have? So already, if we have that, we can now come up with a 16 Punnett square. So what do we have? So we have something like this. Okay, so we have D small b, D small b, D small b, D small b. So we have 4 of them. So first let's get this. So we have our D small letter, so D, big letter B. Then we have our D, small b. Then we have our D, big B. Then last but not the least, we have two small letters, our D, B, like that. Then this side we are going to have our DB, small letters DB, DB, DB. So it's only after you do this Punnett square that's when you can enter there. So our D and our D, so we are going to have D, small letter D, B, small letter B. Then we have our D. So what we have is small letter D, like that. Then we have our B, small b. We go to our D, small d. We have B, small b. We have our D, small d, B, small b. Then we have our D, small d. We have our B, small b, like that. We are here, so we have D, small d, B, small b. We have our D, small d. We have B, small b. We have our D, small d. We have our B, small b, like that. Then we are going to have our D here with D. We have B, small b. We have here our D, small d. We have B, small b, like that. Then here we have our D, small d. We have our B, small b, like that. Then we are going to get a D, small d. We have our B, small b, like that. So last but not the least, we have our D, small d, our B, small b. So we are just using D, small d, B, small b, D, small d, B, small b. We have our D, small d. We have B, small b, like that. So this is what we have for that problem. Now after doing this, we can now come up with the ratios. So we can say to, since big letter D is to stems and hairy stems. We are going to have to stems and hairless stems. Then we have short stems and hairy stems. We have short stems and hairless stems. So we are going to count. So for toe, we should have at least one D. For hairy, we should have at least one big letter D. So according to the question, we are told that hairy stems are produced by the dominant. So hairy stems are produced by the dominant, which is very, very important. They are produced by the dominant B. So we expect at least one big letter D and at least one big letter B, like that. So if we count there, we are still going to have a 4 like that. Then here we are going to have our D, which is here. We should have hairless, we should have two small letter B's, at least one D for toe still. So we are going to have this type of a situation. So we are going to have 4 there. Then here we are going to have short stems but hairy stems. So short has to have two small letter D's. So we have short stems and hairy. So meaning to say we expect to see at least one big letter B but two small letters of D. So we are going to have a 4 there, which will give us this. Then here we are going to have 4 short stems and hairless stems. We expect to have something like this. So we have this, we have something like this. So it will be our 4 like that. So the ratio is a 4 to 4 to 4 to 4. So the phenotypic ratio, which can be reduced to a 1 to 1 to 1 to 1 like that. So this is the outcome for that one. You obtain your 12 marks there. The next question says give 2 examples of each of the following. So if they ask incomplete dominance and codominance, so incomplete dominance, this is where when 2 genotypes are combined, what happens is that the resulting is an intermediate. So we have an intermediate where we have something that is in the middle. No gene is more like dominance over the other, but they are just in the middle. Codominance, on the other hand, occurs when 2 traits or genes are able to show at the same time. Like for example, if let's say we have something like that, if the parents had the traits for red color like that, then at the same time we have yellow like that. If we have yellow, that means to say the offspring produced is showing incomplete dominance because, or is showing codominance because both colors are showing. On the other hand, incomplete dominance occurs when if we have, let's say, red and yellow like that, the offspring produced, we have that let's say the parents were this, then the offspring produced is purple. So since these have blended together to form a neutral color, then that is what we call incomplete dominance. So examples of incomplete dominance that we might have is the famous example of snapdragon. So the snapdragon is a color produced when a white flower is combined with a red color, with red, to produce pink. So this pink is an incomplete dominance because it is an intermediate. It is not the outcome that was expected. The other one we can have in humans, if let's say the mother had curly hair and the father had straight hair, then the outcome is to produce waves. So curls might look like something like this, straight hair like that, then waves come out like that. So the offspring is seen to be more like expressing incomplete dominance because waves. It is not curly, it's not straight hair, but it's forming waves. For the other thing, for codominance, we might have codominance. We can have something like blood group AB, both A and B show. Then for the other example that we can have is roan cow, the cow that is produced when a white cow and a red cow are fused, where we have individual whites and all that. So there are a lot of examples that you can give. So as long as we have patches, we have white, we have red, we have that, then that is called codominance because they are both existing at the same time. This question says, one of the functions of the smooth endoplasmic reticulum is the synthesis of carbohydrates and lipids. Then we are told use the diagram of the animal cell to explain how these products move from the smooth endoplasmic reticulum to the neighboring cells. So we are required to come up with a diagram. So let's draw the animal cell with just a cell membrane like that. So then what are we going to have? Let's increase this. Let's increase the size a bit. So if we have important detail, we are going to say the smooth endoplasmic reticulum like that. Then we have the mitochondria like that. Another important detail we have, let's say we have the Golgi body like that. So we might have the nucleus like that. The other things that we might have are, we might have some cytoplasm and all that. So we can just label some things. We have the cell membrane here. We have the Golgi body. We have the smooth endoplasmic reticulum. We can just draw that. We have the mitochondria like that. So now, to explain how substances move, we are going to say, firstly, when lipids and carbohydrates are, according to what we have been told, are synthesized. So for this question, I'll explain how you are supposed to answer in a paragraph like this, not using points. So you can say when lipids, so firstly, let's have the lipids. So when they are created, they are packaged into what we call transport vesicles. So this, what you see here is a transport vesicle. When it begins to move like that, it will enter into what we call the Golgi apparatus or the Golgi body. So the first thing is that it is packaged in a transport vesicle, which was the one you saw moving. Then it enters the Golgi body for modification. When they are modified, let's say this is the modified structure, then they are also now packaged into what we call secretory vesicles like that. So let's say this is the modified form where they add, let's say, carbohydrates, there is the folding and all that. Then they are packaged into secretory vesicles. So now these will be called secretory vesicles. So this is supposed to be double S, so VESSICO. So after that, it will start to move. So it will start to move like that. So as it moves, so this is the secretory vesicle. It moves, it embeds itself in the cell membrane like that. Then from there, it will release the materials by opening up. So this becomes a part of the cell membrane. So the cell membrane like that. So the cell membrane, yes, it might look this small, but it's something like that. So it embeds itself in the cell membrane like that by entering, or rather by fusing with the cell membrane. So we might have the cell membrane like that. When this was fusing there, it looks like something like this. It enters like that. Then this is more like becoming a part of it. Then after it reaches that point, it opens up like that. When it opens up like that, it will more like secrete those materials outside the extracellular fluid. Extracellular fluid, we are just talking about the fluids that are found in between cells. So this it passes through this extracellular fluid and enters the other cell through various processes such as endocytosis. So the release of these materials, so the excretory vesicle moves there. This is called exocytosis, because there is the release of substances outside the cell. Then when this cell enters another cell, this is what we are going to call endocytosis. So when those materials enter a neighboring cell, they will be called endocytosis. Okay, so this is how you write your essay. So we can say when lipids are synthesized, they are packaged into transport vesicles, just as we said, transport vesicles, and they are moved to the Golgi body for modification. After they have been modified, they are packaged again into secretory vesicles which go outside. These secretory vesicles attach themselves to the cell membrane, and then they release substances through exocytosis to the extracellular fluid, which is just the fluid in between cells. Then finally the materials enter the other cell through various processes such as endocytosis. Let's look at our next question. So we have answered that. Let's go on to our next question, which is states the function of the cytoskeleton. So the cytoskeleton, just what you can think about is it's a skeleton. So already that gives you the function. And you know the function of the skeleton is number one, structural support. Number one. Number two, the cytoskeleton is used to move substances within the cell. Number three, it is used to move the entire cell. So if let's say we have a cell like this, you can just think of the cytoskeleton as number one, a pillar like that. So this is the cytoskeleton. So it holds, it offers structural support. That's number one. Then number two, we are going to have something like this. Number two, it helps in the movement of internal substances. Then number three, it helps in the movement of the entire cell. So as you can see, for materials like that within the cell, they are moved by the cytoskeleton. When you look at your chromosomes, the chromosomes are moved by the cytoskeleton in that manner. So that is it for the cytoskeleton. So it moves substances within the cell. It also helps in the movement of the entire cell. Let's move on. If we have state how molecules are moved from one side of the membrane to the other. So if we have, let's say this is the membrane that we have, then this membrane is moved from one side to the other. So if this is the membrane, let's say this is the inside. If this is the inside, let's say this is the outside. So substances are moved via what we call simple diffusion. That's number one, simple diffusion. So simple diffusion involves the movement of substances. So remember the movement of substances. Simple diffusion. So simple diffusion involves the movement of substances that are small, such as carbon dioxide and water, through the space like that. Then the other diffusion that is there, we have substances. So the movement of substances, you can just write the movement of substances from a region of higher concentration, or from a region of higher concentration to a region of lower concentration. So small substances such as carbon dioxide can pass, oxygen can pass through simple diffusion. Then we have facilitated. So these are some of the ways. So facilitated diffusion. So facilitated diffusion. Under this one, we are going to have facilitated diffusion. We are going to have the mechanism in which substances that mostly dissolve in water, such as glucose and amino acids, and are large, they cannot easily pass through what we call facilitated diffusion. So this one works using the protein channels. So here we can have something like this. So an integrated protein channel like that allows certain substances to pass through. So let's say this is our glucose. So our glucose will be able to pass like that. So this is a facilitated diffusion. So using a membrane, protein integrated in the cell wall like that. So this is the membrane protein integrated in the cell wall to allow certain substances to pass, so it does not require energy. So let's write something on facilitated diffusion. So we can say concentration. So facilitated diffusion involves the movement of substances down a concentration gradient. So from a region, that means from a region of higher to a region of lower concentration towards the down the concentration gradient using a protein channel. So substances such as glucose pass in this way. So mostly substances that dissolve in water pass in this way. If they are big, they will pass in this way. Even water itself passes through the cell membrane using special channels that we call aquaporins. So aquaporins are the channels through which water passes, because already the tails do not like water here, so it is very difficult for water to pass through. Some might pass, but it will be very, very slow. So water uses the same mechanism of using a channel to pass through so that it can. So a channel only allows certain substances. So if it's a water channel, it will only allow water like that. The other means of transport that we have is what we are going to call bulk transport. So bulk transport, when a substance is very, very large, it will pass through the process of bulk transport, which might involve endocytosis, or we might have what we call exocytosis. So just as the name it entails, endo means inside. So endocytosis involves the movements of substances towards the inside of the cell, exocytosis towards the outside of the cell. So substances that are very large, such as bacteria, pass through exocytosis and endocytosis. So this is how the mechanism works. Let's say this is the bacteria, this one is our bacteria like that, then this one is our cell membrane. So endocytosis involves where the cell membrane changes shape. So the cell membrane might change shape like that. So as the bacteria moves closer like that, we might have something like that. So as it reaches that point, this membrane closes up like that. So it will start to close up like that. When it closes up there, let's say, let me just extend the cell membrane. Let's say it's this size. Then it will move to another portion like that. So this is what is called now the food vacuole. So this is what we call the food vacuole, which will move this substance. The food vacuole, you can just finish it up. So we have the food vacuole, which moves substances inside the cell, so until it enters the cell like that. So this is the food vacuole like that. So this is how substances are moved from the cell into the inside. Likewise, even the other case is true. When it reaches this stage, it changes the shape and all that. Substances are moved in and out of the cell in that way. So when they are moved out of the cell, then there is a release here, then to release the substance. Other nutrients such as lipids are moved out of the cell in this manner. That is called exocytosis if it's going outside. If it is going outside, it's exocytosis. If it is coming inside, it is endocytosis. So that is how the mechanism of transport works. So we are here now. States the characteristic of prokaryotic flagellum. So the prokaryote looks like something like this. It looks like something like this. So what is the function of the flagellum? Or rather, what are the characteristics, not functions? What are the characteristics of the flagellum? So number one, the flagellum, what you can just, the characteristics are that, so remember, you're not supposed to write points like here. I showed you how you do the question. You answer like how we answered the first question, which was when this is how you write the paragraph. So here, I can use points so that I am much faster. So we have the flagellum. Flagella is made up of the protein flagellum. So it is made up of the protein flagellum. Then we are going to have it is made up of the protein flagellum. It moves by rotary, which is rotational motion, not wiping. So it rotates. It's more like it makes a circle, and it does not wipe like wiping might be. It goes this side, goes this other side, like that. No, it rotates. So it rotates, forming more like a circle. If it is going, let's say, this direction, then the tail shall rotate like in this direction. So it shall make a circle like that. That is what we mean by rotations. Then the other thing about the flagellum is that it is powered by the proton motive force, which is this force is which is created by the movement of hydrogen ions. So this is hydrogen ions from one side of the membrane to the other. So from one side of the membrane to the other. So as they move in and out, they create this force, not directly ATP, but they create this force. So as let's say if this is the membrane, as hydrogen ions move in and out, they create the proton motive force, which powers the rotation of the flagellum. So those are the characteristics of the prokaryotic flagellum. Let's move on to our next question. It says that compare the processes of independent assortment and crossing over. Which process has the greatest influence? So we must first define what they are. So independent assortment is we are just talking about how chromosomes align themselves independently of each other, in the sense that, let's say, if we have during meiosis 1, let's say we have the metaphase plate like that, so chromosomes might align themselves like that, like that, like that, like that, like that. Now these chromosomes, remember, one is coming from the mother, one is coming from the father. So we might have something like this. So let's say the ones in yellow are from the mother, then the ones in red are from the father. So there's a possibility that chromosomes might align themselves in such a manner. So it is not necessarily that one has to be there. They could reshuffle in as many ways as possible. We could have from the mother, from the father, from the mother, from the mother, and all that. We could still have something like that. So this is done in a random order. So when there is the splitting, that's how come we could have traits. We could have traits, many traits in that manner. So that is random or independent assortment. While crossing over, on the other hand, is what happens when the two chromosomes attach themselves at the chiasmata. So that is the first part. We define the two terms. So so here we have during meiosis, crossing over occurs under prophase 1 when the homologous chromosomes pair up. So they have to pair up, then they exchange genetic information at the chiasmata. Then they exchange segments of DNA at the chiasmata, creating new combinations of alleles on the same chromosomes. Random, which is also known as independent assortment, occurs in meiosis 1 as well. So under this one, the chromosomes align themselves at the metaphase plate side by side like that in a random manner, so that the maternal or the paternal are distributed independently into gametes. So they are independently distributed into gametes. One, the maternal, which is the mother, can be like that. It could be like that, as numerous as that. It's not something that is done systematically like that. So both of these, quite alright, they produce effects, or rather diversity, or influence genetic diversity. But which one has got a greater influence? So already this is already 4 marks. Now this question is a question of intense debate, which you can research more. So the one that I will choose, the one that contributes more to the crossing over, is what we are going to call random assortment. So we can say random assortment carries more impact. So random assortment, the number of possibilities, remember there are 23 pairs, so there are 23 chromosomes. Now the number of possibilities of rearranging these chromosomes, this chromosome might be this side, this might be this side, this might be this side, this might be this side, is number 1. We can have 23 if those were to swap. But there is a possibility that this could swap, but this doesn't swap. But that's also another possibility that will carry another 23. This could swap and this could not swap. So there are a lot of possibilities for random assortment. But remember, both of these are carrying different traits. Then we could have, these can exchange position, this can come here, this other one can come there, and all that. So the number of possible arrangements is a 2 to the power of 23 times in which they can be arranged, which is equal to 8.4 million. So this 8.4 million is the number of different outcomes that can be provided by the random assortment. So just as I said, you can even ask your professor just to be sure, but this is one of the best answers through research, through rigorous research. But others might also argue to say even crossing over, because crossing over can occur at different portions. So for this, I've chosen random assortments, as each carries more impacts, which is about 8.4 million times. So you can just write million times, which is 2 to the power 23 number of arrangements. So random assortment carries more impacts or influences more on genetic diversity, because all these chromosomes have got different, more like characteristics for that gene. So let's move on to our next question. Question 3 states 2 similarities and 2 differences between meiosis 1 and meiosis 2. So what are the similarities and differences? Let's begin with similarities. So the similarities that we might have under meiosis 1 and meiosis 2 is that both of them, meiosis 1 and meiosis 2. So in meiosis 1 and meiosis 2, the difference is that we are going to have, number 1, in both cases, chromosomes align or rather migrate at the metaphase plate, at the metaphase plates like that. Then for the other thing, so though it's a bit different, but they all have to be at the metaphase plate. So meiosis 1, which is this one, meiosis 1, then meiosis 2, they also go at the metaphase plates, though not side by side, it's just one line like that. Then the other thing that is done is that there is the disintegration of the nucleus. So the nucleus, which is the nuclear pore and the nuclear envelope, disintegrates. The other thing that occurs is what we are going to call cytokinesis. Cytokinesis is simply the splitting or the division of the cytoplasm. So the division of the cytoplasm. Then on the differences that we might have, the differences that we have is number one, we can talk about DNA replication. So DNA replication only occurs in meiosis 1. So in meiosis 1, DNA replication occurs. There is the replication of DNA and all that. So it's not necessarily like DNA replication occurs in meiosis 1, but it occurs in S phase of interphase. Then after interphase, we are going to have meiosis 1. But in meiosis 2, there is no DNA replication. It does not undergo the interphase, but it after meiosis 1, there is a brief stage which we call interkinesis, which is a rest between cell cycles from meiosis 1 to meiosis 2. So meiosis 1 is preceded, so it is preceded by DNA replication, meaning to say first DNA replication takes place, then meiosis 1 will take place. But for meiosis 2, there is no preceding of DNA replication. So precede simply means before it occurs, there is DNA. Before it occurs, the process, before it occurs, there should be DNA replication. The other thing that is different is that under meiosis 1, we have what we call homologous pairing. So there is the pairing of homologous chromosomes. So homologous chromosomes might be like this, so they might be paired like that. So this is what we call the homologous chromosomes pair up like that. While for this other one, what we have is there is no homologous pairing under meiosis 2. So take note. So meiosis 1, so I can just write 1, there is homologous pairing, whilst meiosis 2, there is no homologous pairing. So in meiosis 1, they will exist as a pair like that. The other difference is that under meiosis 1, I will just write under meiosis 1, there is a reduction of the ploid state from diploid. There is the reduction, you can write there is a reduction from diploid to haploid. So we have, for example, if you look at how the cells are arranged like that, when they are moved apart like that, we are going to end up with cells that have been moved apart in different directions. When the cell is cut like that, then we are going to have half the number of chromosomes. So this is the reduction of the state from diploids to haploids. That is under meiosis 1, like that. Now meiosis 2, the haploid, whilst in meiosis 2, haploid state is maintained. So meiosis 2, the haploid state is maintained. So under meiosis 2, they are just put apart. We can have, so this cell has to divide into 2. So if we have this structure like that, it is broken down to more like release, like that. It is just, the chromosome is just split, but still we have 2 chromosomes. So even if chromosomes are attached together like that, they are called, we are just going to call them as 2 chromosomes. Even these are 2 chromosomes. So the haploid state is maintained like that under meiosis 2. Let's move on. This one says states the similarities and not this one, state 4 stages of prokaryotic cell division. So prokaryotic cell division involves, so stating, we can use points here since it's stating. So prokaryotic cell division involves cell elongation, which is cell growth. After cell growth or cell elongation, there will be DNA replication. After DNA replication, there is DNA segregation, meaning to say DNA is moved to opposite poles. So DNA segregation, to segregate, is to move things from one side to the other in opposite direction, should I say. Then we also have the process of septation, which is just the formation of a septum. If you want more information about this, so the septum, which forms the zeroing at the middle, which helps more like constrict the cell, thereby dividing it into 2. If you need more of these videos, all these are available. Then last but not the least, we also have cell separation by what we are going to call cytokinesis. So the cytokinesis involves the constriction of the cell wall, in which the cell wall pinches and then it separates the cytoplasm into 2. So how many were we given? State 4 stages. So we can even combine septation, not just septum, septation or septum formation, septation and cytokinesis. We can combine it in that manner. For this one, it's saying that what would happen if meiosis, if the spindle checkpoint is dysfunctional. So now, what is the spindle checkpoint? So the spindle checkpoint, it is the point at which the chromosomes are checked whether or not the spindle fibers are correctly attached. So if we have something like this, we have something like that. So if the spindle fibers are correctly attached. So now, if the spindle checkpoint is dysfunctional, this could happen. So dysfunctional means it's not working properly. So this could happen. Now the problem with this is that chromosomes will be split. Now after the spindle checkpoint, the cycle will bypass will bypass metaphase, in which there is the attachments there. Now the problem is that when it bypasses this cycle, it will move on to what we are going to call to anaphase, in which we have chromosomes like that, then this side will have chromosomes like that, and a complete thing like that. Then when it is split, it will be a chromosome like that, another chromosome like that, and a full thing like that. Now this is a problem. This condition is what we are going now to call aneuploidy. So aneuploidy is when there is an even number of chromosomes. This is a problem as it leads to a potential mutation. So that is the effect if the spindle checkpoint is dysfunctional. Let me write something here. So just as we have said, if the spindle checkpoint is dysfunctional, that means to say it's not working. The cell will bypass the M phase, which is the checkpoint phase, and proceed to anaphase. Anaphase, this is where they are pulled apart. Now, even if chromosomes are not properly attached to the spindle fibers, there will be the splitting. Now, if not all chromosomes are attached to the spindle fibers, then there will be a situation where we call disjunction. So know these terms. Disjunction simply means there is an even distribution, or rather the unusual splitting of chromosomes where one side has less. This condition is what we call aneuploidy, when there is an even distribution of chromosomes than the required number. Now, if let's say this chromosome has got 24 chromosomes, then it fuses with the one with 23. If you add these up, you're going to have 47 chromosomes. This leads to the creation of Down syndrome, which is trisomy 21. When we have 23 chromosomes for the 21, we are going to have trisomy, which is Down syndrome. Or we might have monosomy, where someone has less number of chromosomes. Like if one has got 22, the other one has got 23, when they add up, they make 45. We can have situations such as the Turner syndrome. Let's move on to the next question. So this is, mules are offsprings of mating of horse and donkey.
What? Mule.
Mules are unable to reproduce. A horse has 44 chromosomes, whereas a donkey has 42. Use your knowledge of meiosis to predict the diploid chromosome number of mule. So we are going to have the horse has got 44, then the donkey has got 42. So the haploid state of the horse, meaning to say haploid, we divide by 2, which will be a 22. Then even here we divide by 2, we are going to have a 21, like that. Therefore, haploid diploid state of mule, so even this one is haploid of the donkey, meaning we divide by 2. Then diploid state, it means to say we have to add the horse and the donkey, which will be our 22 plus our 21, which will give us a 43 number of chromosomes. So that's how we answer that one. Then when we look at based on your answer in A1 above, propose a possible explanation for an inability of mule to reproduce. So for this one, what we are going to have is that so now what could so let me read the question again. Based on your answer in I above, propose a possible explanation for the inability of mule to reproduce. So mule cannot reproduce because number one, so if we have odd number of chromosomes, odd chromosome number, not all of them are able to form homologous pairs. So as you can see, not all of them are able to form homologous pairs like that. Then as you know, the homologous pairs, let's say if we have number 4, we can have something like this. So these are pairs. So if we have 4 chromosomes, they can form 2 pairs like that. But since Mu has got, let's say 43 is an odd number, not all of them have a pair, and that would cause Mu's inability to reproduce. So that's number 1. Number 2, due to the fact that there is an odd number, there is also what we are going to call failure of synapsis. So synapsis is similar to just the alignment. So synapsis is just a fancy way of saying that the 2 chromosomes align themselves together like that. So now, this is a build-up knowledge. So not all of them are able to form homologous pairs. If they cannot be able to form homologous pairs, that means to say even there will be failure of synapsis. Synapsis, which is when they have completely paired up like that. Now, if there is no synapsis already, that means to say these will fail to more like move at the equator side by side like that. So due to that factor, Mu is unable to reproduce. So all these, the failure to form homologous chromosomes and failure of synapsis, thereby preventing the chromosomes from aligning at the metaphase plate. You should know that the failure of the chromosomes to align evenly at the metaphase plate causes the total failure of meiosis, which renders Mu incapable of reproducing. So we are here. It says that explain the importance of G1S checkpoint. So G1S checkpoint of the cell cycle is when the cell is about to go into S phase, which is the synthesis stage. So why is it important? So G1 makes sure that the importance of G1 is that it makes sure that there is nutritional sufficiency. That's number 1. There is nutritional sufficiency. Number 2, it also makes sure that DNA is not damaged. So in other words, the G1S checkpoint is very, very important as it halts, which halts means halts or stops the cell cycle from proceeding if these conditions are not met. This is very, very important as we do not want an immature cell from reproducing, and also it makes sure that DNA is not damaged. If it is damaged, the cycle is halted or stopped or put on hold so that it can be repaired, or if the cell can just be destroyed so that there is no passing down genetic information to the offspring, 40 offspring. So that's that. Let's look at our last sets of questions, which is our question 4. So we are here using molecular structures. Describe how the following are joined during polymerization. So the molecular structure of proteins. So we have amino acids. You can watch that video. So amino acids will look like this. Then we have something like this. We have our OH, we have double bond O, we have H, we have our R group there. Then we have our N, we have our H, we have our H like that. Then if we combine this with another one, we are going to get a. So now when these two combine, they will have something like this. So this whole thing will be taken like that to release water, because this is a dehydration reaction, to form C double bond O there, C, our H, we have our R, we have our N, we have our H, we have our H, we have. So since this has been taken, this will bond with nitrogen. Then we have H, we have our C, we have our H, we have our R, we have our C double bond O, we have our OH. So this is how the reaction takes place. So to describe this, we are going to say, so this is what we have. So proteins are formed when the carboxyl group, which is the one with our C, this one, so just as you are seeing, this one reacts with an amino group, which is this group. When it reacts with an amino group through a condensation reaction involves the removal of water. This eliminates water molecules. To create a covalent peptide bond, linking the monomers into a peptide chain. So the type of bonds that is there is a peptide bond. In case you are required to identify which one is the peptide bond, this is the one. So this is the peptide bond, like that. So let me just color it a bit. So this is the peptide bond there. Oh, sorry, not there. Here, where the reaction took place. So the reaction took place here. So this is the peptide bond. So for that one, it was saying using the molecular structure. So we draw like that, then we add a statement. Then for nucleic acids, we have, for nucleic acids, this is the structure that is there. So we are going to have a sugar, which is going to look like this. So if you need more information on this, you can just watch some videos which I have created. So we have something like this. We have the nitrogenous base. Then we are going to have our other thing here. So we have our O there. Then we have our P. We have our O. We have our O. Then we have our O like that. This O joined together, joins another carbon like that. Yeah, so like that. In which case, we are going to have something like this. So another carbon, we have another group like that. So if you want to know how these come about, you can just watch or comment in the description link, so that I can be able to forward the link for you. So we have something like this. So the structure continues like that. So even here where we have our CH2, it can continue where we have our O, we have our P, we have our O, we have our O like that. The structure continues like that. OH if there is no other structure on top there. So this is basically how nucleic acids look. So depending on the nucleic acids, it could be a deoxyribose for a sugar here, which is hydrogen there. Then here we have our hydrogen like that. So if you want to know how this structure comes about, just comment so that I can forward the video. So the description of how they fuse. So as you can see here, this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, this is the fifth carbon. So that's how we count primes. So from oxygen, where there is a bend, that's number 1, number 2, number 3, number 4, number 5. So 5 prime. So as you can see, the joining is happening at the 3 prime and at the 5 prime. So then the bond that is formed, this is held together by what we call a phosphodiester bond. So this phosphodiester bond is a bond formed by a phosphate group like that. So this phosphate group helps in holding the two bonds together. So to define what a phosphodiester bond is, you can just say a phosphodiester bond is a bond that holds two groups of nucleosides together. So two groups of nucleosides together, that's the phosphodiester bond, or should I say two groups of nucleotides together forms what we call a phosphodiester bond. So on the other hand, so we can define it as a nucleic acids are formed when a phosphate group of a 5' carbon, so 5' meaning this position, when the phosphate joins with a 5' carbon of a nucleotide reacts with an hydroxyl of a 3' carbon. So the 3' is here. So here there is an hydroxyl. When they react there, they are going to form. So if you need more information about this, just comment on the 5' carbon of a nucleotide reacts with an hydroxyl at the 3' carbon. 3' carbon of another. This condensation reaction forms what we are going to call a phosphodiester bond. So a bond made of phosphates that holds two nucleotides together is a phosphodiester bond, creating a sugar phosphate backbone with a distinct 5' to 3' directionally. So we are just talking about the 5', and if this is 5', automatically this other part will be if this is 3', this is a 5' here. So that is what we simply mean. So just remaining with a few questions, let's look at our next one. Give a detailed description of globular proteins. So globular proteins, so these proteins are, number one, just from the word globular, just think of a sphere. So globular proteins are spherical. They are spherical compact. So they are spherical and compact. Then you can add an example. So e.g., so most of them, so what is the question description? And are mostly soluble in water. So the example, so this is the description. The example that we might have is enzymes. We might have hormones such as insulin, among other things. Let's move on to the difference between the leading and lagging strand. So leading and lagging strand. So the leading and lagging strand, let's say if the DNA is being opened in this direction. So the structure of the DNA, if you recall, it looks like this. Now let's say if the DNA is being opened in that direction, what happens? So if the DNA is being opened like that, you will find that there will be, number one prime here. So the DNA proceeds from the 3' to 5' always. But here, if we have 5 there, we should have a 3', then we are going to have a 5' like that. Then we have this primer will be attached at this point. So if you want more information about this, just comment. All the videos are available. So this means to say it always begins from the 3' going in this direction. So the one that is going in the direction where the DNA is being opened is what we call the leading strand. Then this one, since this is 3', it will be going in the other direction. Now, due to the fact that it is going in the other direction, okay, let's say the primer was put here, let's say the 3' is here for the sake of explanation. If the DNA is going that direction, you will find that there will be some space in that regard. Now, as a result, there will be another addition of a primer, which in the video in another video I have explained, which will move like that. Now, this creates some spaces which we are going to be calling, so it only touches some parts to create the DNA. Now, these are what we are going to call Okazaki fragments. There is space in between. There is space like that. So the leading strand goes in the direction where the DNA is being unwinded. Then for the lagging strand, it goes in the opposite direction like that. So that is the main difference. But you have to mention the 3', 5', and what is happening. So this is the explanation. So the leading strand is a new DNA strand synthesized continually in the 5' to 3'. So what we mean is that if this is 3', that's how the numbers interchange. This shall be 5', this shall be 3'. So it is the strand synthesized continually in the 5' to 3' direction towards the replication fork on the 3' to 5' template. So it's moving from the 3' to the 5'. It's moving like that. So the key thing is that, number one, it only requires a single primer, like this thing that we have put here is a primer, to just continue. It will be able to form the complete DNA with just one thing continuing like that. But for the lagging strand, it needs multiple primers. It puts here, it starts, it puts here. So a primer will indicate where the DNA, the direction of replication should be. So we have the other thing is that whereas the lagging strand is synthesized discontinually in the 5' to 3' direction. Now, even here it is still 5' to 3', but the direction is away from the replication fork where it is being unwinded. It's going in the other direction, though it is still in the 5'. It's starting, if this is 3, this is 5', it's going to the 3', but in the other direction of the replication fork. Now the template, this template forms short segments called Okazaki fragments. So these Okazaki fragments are more like spaces. If you have something like this, if you have the DNA there, then the space, we have the DNA there, then the space is what we are going to call the Okazaki fragment. Not the space, sorry. This is what we are going to call the Okazaki fragments. So that is that. So this requires multiple primers. You need to put one here, you put another one, it continues. But for this one, it is just continuous. So take note of the terms. Leading strand is continuous, while lagging strand is discontinuous. So that's that on leading and lagging strand. So looking at this term, what are the differences between lipogenesis and lipogenesis or lipolysis? So lipo means fat, genesis means to make. So in simpler terms, we can just say lipogenesis is the formation. It is the formation of fats. It is the formation of fats using glucose. So glucose is used to make fats. So the formation of fats, or rather lipids, using glucose is called lipolysis or lipogenesis. Then lipo again means fat, lysis means to break. So whilst lipolysis is the breakdown, it is the breakdown of lipids to fatty acids and glycerols. So from fatty acids and glycerols. Let's look at our D so that we remain with one question there. So our D says the molecular formula for glucose is C6H12O6. Then we are asked what would be the molecular formula for a polymer made by linking 9 glucose molecules together by a dehydration reaction. So a dehydration reaction causes a decrease in the amount of, it's more like water is released. So if we have 9 of them, we are going to put a 9 there. Then we are going to multiply 9 times 6. We are going to have 6. 9 times 6, this will give us a 54. 12 times 9, this will give us 108. Then we have our 6 times 9, it will give us a 54, like that. So this is the glucose that is expected. Now we have to account for the water lost. So we have glucose, so we can write 6 of them. So we have C6H12O6, like that. So we need 9 of them since they are the 9 of them. So the number they are, that's the number you use. So we could have 9 of them like that. So we are going to get something like, for each one there will be water lost, like that. As long as they are close to each other, water lost there. We are going to have water lost. We are going to have water lost. We are going to have our water lost, like that. Even this one combines with this other one, so it's only space, but they will combine again. Water shall be lost there, so you can just put water lost. Then even here we are going to have water lost. Now count the amount of water lost. So we have 1, 2, 3, we have 4, 5, 6, 7, 8, like that. So that means to say, if you have 9, that means to say you will lose 8 moles of water. If you have 7, you will lose 6 moles of water. So what we are going to do is that we are going to get our 6 moles of water here and do this. So the 6 moles of water, we are going to get 6 moles of water lost, like that. Then we are going to multiply 6 times 2, that will give us a 12. Then we have our O6, like that. So we are going to subtract. So remember, I have C54, H108, then we have our O54. So we subtract minus our H12, then we have our O6, like that. So if we subtract, we are just subtracting from hydrogen. I can even put it here. So I can even put it here, which will give me my minus H12O6, like that. So getting this, I'm going to have, why am I having 6? It's not 6 moles of water lost, but it's 8. So we have 8. So here we should have 16. Then here we should have 8. So we have H16 or 6 or 9 or 8. So we are going to subtract our 54 minus 9. This will give us, so 54 minus 9, that will give us, we are going to have O46. Then here 108 minus our 16, this will give us a value of 2. We have 9. So we are going to have our H92, then we have our C54, like that. So this is the molecular formula of that compound. Last question is saying that we have given this DNA strand, write down its complementary RNA and DNA. So we are going to have, so for DNA, this is how we write. So for DNA, we have our C, our G, our A, our T, C, G, A, T, CAT like that. We have 3 prime. Then here we have our 5 prime like that. Then what we are going to have is for DNA, remember the mnemonic that we were using. So we have 3 prime. If we have 5 here, we should exchange. If we have 3 there, we have to put a 5. So the mnemonic is so C bonds with G, so which is the cars in the garage for DNA. Then apple, which is your A, apples on a tree, so A bonds with T. So we are going to have, if we have C there, we are going to put C, it will be cars in the garage, cars in the garage, apples on a tree, apples on a tree, cars in the garage, cars in the garage, so C with G, apples on a tree, cars, apples on a tree. Then we have our C bonds with cars in the garage, apples on a tree, it's on this space. Then we have our apples on a tree like that. Just put this to show where they are being bonded like that. So this is how we draw those structures for DNA. You get your 3 marks. For RNA, it is the same, but it's going to change slightly. So same thing, the numbers will change. We have 3 prime there, we have our 5 prime there. So we are going to get, for RNA, we still have the cars in the garage, but A, it shall be apples under the tree, which is your uracil. Then we are going to have here, we are going to have we have apples on a tree, but this one remains if the T is on top. Then we have cars in the garage, then A under the tree, that's for RNA. We have apples on a tree, cars in the garage, apples on a tree, or under the tree, since it's RNA. Then we are going to have our T, it shall bond with apples on a tree. So we are going to do this. That's how you obtain your 6 marks. So that is how we go about that one. Thank you very much for watching. Hope you would like, subscribe, and share for more. Don't forget to comment each and everything that you want, and don't forget to click that subscribe button. See you in the next tutorial.